Hello, my name is Shane Dunn. This is my glycolysis, gluconeogenesis, and oral aldolase presentation for my Chem 530 class. Real quick, if you are one of my students for my other classes, please disregard this video. It won't be very helpful for you if you're my chemistry students, if you're my biology students. This might be more beneficial, but this is not really the way they will teach it at GCU. So let's begin. First things first, let's go over a kind of recap on what cellular respiration is. So cellular respiration is when you take oxygen and some type of high energy substance such as a glucose molecule, fructose, sucrose, on a carbohydrate molecule, and through a chemical reaction, you make energy. This differs from metabolism because metabolism as a whole is both the catabolic and the anabolic reaction. Catabolic, you can, even though most biology teachers would hate this example, you think like a cannibal. You take a whole person, you break them down and eat them, you cook them and eat them. You take a large molecule, a high energy molecule, and you break it down and you release energy. That is what usually we're going to talk about is mainly cellular respiration is. However, the exact opposite, anabolism, is when you take a bunch of small objects, add them together with an investment of energy to make a large molecule. Metabolism is both, cellular respiration is only the catabolic side. You can have really interesting products through cellular respiration, such as like alcohol, CO2, and water. If you are making alcohol cellular respiration, you are using mainly yeast and its fermentation for alcohol and stuff like that. Many students tend to get that wrong. I think it's funny because if people actually did do fermentation, exercise would be very, very popular than it is now. Over here, we have fermentation. So that's that, or you can go through cellular respiration all the way to the oxidative phosphorylation and the electric transfer chain in between the mitochondrial walls. But we're not going to go that far today. We're just going to talk about glycolysis and gluconeogenesis. So what is glycolysis? Well, glycolysis, you break this word down, which I like to break these convoluted words down into the Latin roots and meanings to make it easier to understand. So glucose just means sugar, glucose sugar, it's a type of sugar, and lysis means break down. So glucolysis or glycolysis is the breakdown of glucose. And it's a catabolic reaction. It is usually a 10-step chemical reaction. There are different pathways. Most of the time it's the Enden-Marhof bonds or EMP pathway is what we mainly learned in schools, what we're going to talk about today. Other pathways do exist though. Glycolysis mainly takes place in the cytosol. So every single cell can do this. You don't need a mitochondria to do this, which means bacteria tend to do this a lot, so do yeast. It's highly exergonic, which means tons of energy released. Well, not tons, but more than given invested. And it's anaerobic. So ana just means without, and then aerobic means air. So you use process without air. There are also two key phrases we'll talk about. Over here on this thing, we have a Fisher projection of what glycolysis looks like, and then we can have the normal chair diagrams here and there. I don't like chair diagrams, I prefer Fisher projections, but there we go. So let's talk about the steps. I was originally going to write each step, but I realized that I have a mouse and pen and that would take too long and kind of boring. So I'm going to go over this easily. I'm going to show you an easy way to how I memorize the steps of glycolysis and break down each way. You notice the enzymes aren't there. I'm going to tell you the enzymes because they're easy ways of knowing what each enzyme does. So first things first is we have glycolysis. Glycolysis is a 6-carbon sugar. So we're just going to draw 6 carbons. 1, 2, 3, 4, 5, 6. So you start with a 6-carbon sugar, and we're going to see what happens to that. The first enzyme is something called hexokinase, hexokinase. And real quick, this is a kinase molecule. So break that word down. That ace right there means it's an enzyme. And then kine means it is a phosphorylating enzyme. So we're going to add a phosphate. And that's how we get glucose 6P or glucose 6-phosphate. And what glucose 6-phosphate looks like is that looks like 1, 2, 3, 4, 5, 6, and then a phosphate at the end. So this right here is glucose 1, this is glucose 6. And that's what the P attaches, that 6 one right there. Now we have glucose 6-phosphate. We're going to have to, don't worry about this right here. These are not necessary right there. After that, we have glucose 6-phosphate. We're going to do something fun. We're going to take this, and the enzyme is called a phospho, because it's going to mess with the phosphate group. All right, phospho, trace word down. Glucose, that's the sugar we're doing. So only going to work with glucose. And then it's isomerase. So, which means, and then isomer, then ACE. Sorry, my dyslexia kicks in. I got to make sure I say these words correctly. Isomerase. So iso means same, and it's an enzyme. So it's an enzyme that's going to keep the same number of carbons, but we're going to rearrange them in a way. Don't worry too much about the way that it's going to work in a chemical way. Just know we took glucose, and now we have fructose 6-phosphate. It's the same number of carbons. It's still 6 carbons with a phosphate on the 6th carbon. It's just the way they rearrange the alcohol groups, the hydrogens and oxygens, are moved around just a little bit, but it's still the same type of molecule. After we have glucose 6-phosphate, we're going to have right here, technically, glucose, sorry, this right here, technically, my bad, goes here. That's my bad. Right, what enzyme that goes right here is going to, oops, my bad, that crossed one hair out. We're only going down right now. This is going to be phospho. So we're doing with phosphate again, right? Fructose. Now we're doing the fructose sugar, right? And then kinase. So now we added 2 phosphates. The kinase can add or remove a phosphate. So now it's the exact same molecule, but now we have 2 phosphates. 1, 6. So now we have a phosphate on the 1 and a phosphate on the 6. So technically it's 1,6-bi. Bi means 2 phosphate, fructose biphosphate, depending on how you say it. Next, what's going to happen is after we do this fructose, is we're actually going to cleave it in half. So when you use this thing here, we're going to take 3, 3, and we're going to cleave it half like this. And what it looks like, it's the exact same group almost, except the phosphate on the side. But in reality, it's the same molecule. And that is done by an enzyme called aldolase. And that just cuts the phosphate in half. Now what happens, we have glycerate, or glycolaldehyde 3P. I just call it G3P. Most teachers will, but it's glyceraldehyde. And what this is it's this, we have 2 of them, right? Because we went from a 6-carbon chain, right? 6 carbons, and we're down to 3 carbons. We have 2 3-carbon molecules. There was 1 6-carbon molecule. Now we have 2 3-carbon molecules. I'll say it again, sorry. We started off with 1 6-carbon molecule, and then we broke that down to 2 3-carbon molecules. And now we're going to manipulate all that, right? And then now we're right here, we're gonna have G3P, dehydrogenase, right? Dehydrogenase. Well, what's that mean, right? Dehydrogenase, put that down. We're just going to take the hydrogen off. So now we have this excess hydrogen flowing around. And what that's going to do, that excess hydrogen is going to basically store the energy up, because the human body doesn't like to lose things or waste molecules. So we're going to take an NADA plus molecule, and we're going to convert that with that hydrogen we just got from dehydrogenase into an NADH molecule. One second, something on. Sorry about that. Something came up during my presentation. So now that occurred, right? That occurs. We move something around, and now we have 1,3-biphosphate, right? Well, we didn't add a phosphate. We're just moving things around, correct? So we still have this one molecule split up. Now we have this. what this looks like is going to be 1,3. So here's a phosphate. We have 3 carbons with that. Going from 1,3 diphosphate glycerate, going down, right? We're going to have phosphoglycerate kinase, right? There's that kinase again, right? Phospho, so now we're going to mess with phosphate, right? Glycerate, that's the molecule we're dealing with, right? Glycerate. Do apologize, I am writing with a pen. My touch screen broke, so I have to write the pen. It's kind of hard when I have teaching and then a class every week and stuff like that. So kinase, so it means we're removing a phosphate, which is why we went from 1,3 biphosphate, diphosphate glycerate to 3 phosphates. Now we just lost one of those phosphates. Where'd that phosphate go? Well, we went from an ADP and we gave that phosphate to an ATP. Right? Remember, we have 2 of these 3 molecules. So in theory, we actually make 2 because we're going to do this twice, 1 for each G3P molecule we did, right? And then right here, we have this molecule right here, that's G3P. And then now we're going to go to phosphoglycerate, right? And then we're going to go to, it's a maltase. So we're just moving the phosphate. Now it's 2 phosphoglycerate, which just means we took the phosphate on that 3 and we just moved it on the 2. Right? And then right here, we have an enzyme called Enlose, and ENOAAS. And then we're going to have, technically, we would just move the phosphate back where it was, and rearrange a couple things. And then now we have pyruvate kinase. And then we lose that last thing. So that kinase gets rid of the last one, and our last thing we make is pyruvate. And we do that twice. So the reason I did it in 2 different colors is 2 different phases. The red phase is called the investment phase, right? So hexokinase and right down here, this phosphofructose kinase, those take, they can be right here, those are going to take an ATP molecule, adenosine triphosphate, which is basically a high energy molecule in the body, which we can think of as the body's currency or money. Anytime we use that, remember, we technically take that idea and we're going to use it to make energy. So we're going to take the ATP and we're going to make ADP. So we broke the phosphate off, releasing energy to make this go, to activate these enzymes. Same thing here. All right, an ADP. So technically, it took, you can think of $2 or 2 ATP molecules to get this process going. But remember, we made technically, it's also times 2. Technically, we made a total of 4 ATP. So this is why, very carefully, when you learn this topic, we talk about net profit, right? Net means total gain. So technically, how much ATP we made? Well, we technically made 4 ATP molecules, right? But it also took 2, and it took 2 investments, so minus 2 ATP, right? So in total, what happens is we only made 2 ATP molecules. This is why glycolysis is an example of an exergonic reaction, a catabolic reaction. We took a 6-carbon molecule, broke it down to 3 carbons, because pyruvate is just 3 carbons. And we made energy from, we made 2 ATP. So net profit, we made money. Glycolysis, good job. This is why a lot of yeast, bacteria, our normal animal cells, plant cells kind of do something like this. Plant cells don't, they do something different. You know that part. But they also technically make 2 other high energy molecules, NADH, which we'll talk about when we talk about electric transport chain, Krebs cycle, and stuff like that. Well, that's the glycolysis steps. And then you pause the video if you need to write this stuff down. So That's what glycolysis is. Well, this is gluconeogenesis. Well, gluco means glucose again, so that's sugar. Neo means new, and genesis means creation, like in the Bible. So this is the formation of a new glucose molecule, but it comes from a non-carbohydrate source. What's that mean? Well, I have candy right here because I'm studying. I need energy, right? So I take that piece of my candy and I eat it, and my body takes that fructose and sucrose and glucose and breaks it down and does glycolysis. That is glycolysis. Gluconeogenesis is I take some other substance that wasn't a high energy molecule, and then I make glucose from that. So what can we do that with? Well, we can actually take this pyruvate molecule and we can actually go backwards. And that's the process I'm going to talk about. But you can also do the same thing with glucogenic or amino acids. So anytime amino acid that can be broken down in the glucose cycle, but you can take amino acid, a protein, and make energy from it and use glycolysis from it. You can take triglycerides, a fatty molecule, and do it with that. And you can take lactate, which is pyruvate working out the lactic acid. So the lactic acid make it pyruvate and work backwards. So technically, it's the exact opposite of glucose. So it's an anabolic reaction. It's going to technically take energy. That's why it's endergonic. Taking these non-energy efficient molecules and investing tons of energy into them to make them an energy efficient molecule, high energy molecule like glucose. So why would we do this? Well, one, like I said, the body hates wasting things, right? So what are we going to do? Well, let's say, for example, we didn't go to the THA cycle or the electron transport chain, and we just have a ton of pyruvate or lactic acid around. The body's not going to sit there and waste that. It's going to want to make that into glucose to try doing that again to make more ATP, right? So does that as well. It also helps maintain blood sugar levels, right? Well, if your body needs glucose, might as well not waste it and make glucose and store it when you need it. This is mainly done in the liver and kidneys, mainly in the liver though. Kidneys a little bit, liver is what mainly does this stuff. So, whoa, see, now its arrows are going back up. So I got to talk about a concept real quickly. A lot of you guys in chemistry are gonna know this. That is the concept of Gibbs free energy, or delta G. So delta G is free energy. So what does that mean? We'll talk about the concept of free energy. So if energy is free, it's really good to do that, right? So it doesn't take much energy to sit on the couch and watch Netflix and just relax and not do your work, right? It takes energy, it takes investment or our energy to do work, right? What would you rather do? Study and do your work, or sit on the couch and do nothing? Sit on the couch and do nothing. Our body's the same way. It's lazy. It's going to do the most energy efficient way. So if you have negative delta G is Gibbs free energy, that is free, available. You make more energy than you use, right? Enzymes only want to go in a place that makes the least amount of energy. So when I'm talking about this, is a lot of gluconeogenesis enzymes are exactly the same, except a handful, because those processes need to go in a more energy efficient way. It would take way too much more energy to go backwards. Remember, this is an endergonic. We are investing energy, catabolic, endergonic energy. So we wanted to invest energy, but we don't invest way too much more energy than we're bringing out, right? So first things first is we have this pyruvate carbolaxase, basically. And we're into that pyruvate. We're going to invest 2 ATP molecules into that, right? Boom, invest 2 ATP to get this process going. So it takes a lot of investment to get this going. And then we have oxaloxetate. What the hell is that? That is just a thing, it's not going to last. And then we're going to invest this GTP. What is GTP? ATP, that is like the grade A Energizer Bunny battery. GTP, dollar store battery, does give some energy, not as much. So basically, let's think about this. We invested $2 American and like 2 Mexican pesos, basically. But we're investing way more energy right now to get this process going. But once we do, it gets going. So 2 ATP in this process right here. So we lost 2 ATP and then we lost 2 GDP, right? Process going here. Now, hey, we're back to this this, right? Oh, that's just this same thing. We've seen it before, right there, correct? And we know exactly what enzyme does this, right? That's just enolose. E-N-O. Yep, and lace, enolase. Boom, right? Oh, what happens here? Well, that's just the phosphomaltase, right? Hey, it's the same thing. It's the exact same maltase, right? It's the exact same enzymes, right? Hey, look, right after maltase, right? We have phosphoglycerate kinase, right? And then after the kinase, we have the GC3 dehydrogenase, correct? So the exact same processes here and here. I'm not going to write them down, just save some time, because I really want to focus on the ones that are different, which I'll do in red. So this one is different, and technically phosphoglycerate carbonoxase is different, right? Whoa, what's going on here, right? This is a different one. This right here is now we're going from glyceraldehyde or G3P back to fructose. It's not the same process. Technically, that is fructose, because now we're going to make fructose. Fructose, right? Fructose 1,6 biphosphates. Phosphates. It's very hard to write with a mouse on a crowded desk. I do apologize. And then phosphatase is an enzyme. Basically, it's going to take the G3P and make that phosphate, right? And then boom, 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 say exact same enzymes, except right here, we have this glucose-6-phosphatase. It turns glucose 6-phosphate into that, right? Exact same enzymes, going backwards and forwards. The one I want to keep in function, though, because it's the main thing on this presentation, is this enzyme right here, aldolase. Exact same one, but we're talking about that guy in a little bit. So just know that he's both there. So let's see these steps side by side, basically. So here's your side by side. Red going down is glucose, catabolic, energy release. Blue going up, and androgonic, anabolic, energy is being stored, right? It's highlighting pink. That guy is special, that guy is special, and that guy is special, and that guy is special, right? So the red molecules, the red enzymes only appear in glycolysis. The blue molecules only appear in gluconeogenesis, right? We got this guy right here, aldolase. He helps in both. So he's very, very important. He also breaks the glucose down. So he's a key part. So let's talk about him a little bit. So aldolase, right? It's an enzyme. It's aldolase, sorry, not aldolase. Yeah, aldolase. Sorry, it's making kind of cool fast. I'm worried about the timer. It's an enzyme. So an enzyme is basically a protein that's going to catalyze the reverse. You saw it go from glycolysis going down and gluconeogenesis going up. So it catalyzes both sides. It's a reversible enzyme. It goes from fructose 1,6 biphosphate to G3P, or G3P to fructose 1,6 biphosphate. Goes both ways. And interesting, this enzyme actually has three isoenzymes, or three isospin same, so three enzymes that are pretty much the exact same thing, but differ a little bit in function. Aldolase A, B, and C. Aldolase A, this is kind of interesting. It is on chromosome 16. It's a gene that's on chromosome 16. It is highly found in growing embryo and in muscles, in high amount of muscles, right? It's not found in the liver, kidneys, or the intestines. And kind of found a little bit in the brain, but just know for aldolase A, embryos and muscles where it's found. Well, why? When you work out and do bicep curls or push-ups or exercise, you're using energy. How do you get energy to move your muscle, right? This takes ATP to move my muscle and stuff like that. It takes energy to breathe. Well, glycolysis, Krebs cycle, oxidative phosphorylation, electron transport chain, all that happens. Aldolase is needed for that. Remember, aldolase takes your glucose 1,6 phosphates and makes it G3P. Takes that 6-carbon molecule and turns it to 3-carbon molecule. That's what does it, is aldolase. That's why it's highly important in muscles, because muscles need a ton of ATP. Heart needs to beat every single second. You're constantly using your muscles right now to sit up straight or lay down. That takes muscles. If you had no muscle moving, things would be bad with you happening. If you, for example, there are deficiencies. If you do not have high amount of aldolase A, you have this thing called myopathy, which is muscle weakness. You can't physically move your muscles and they waste away because you're not using them because of this. You also can have hemolytic anemia. You do not have the energy to make red blood cells. Red blood cells don't get that nice glucose. They don't get energy to be made. That's a waste of time if you're not transporting glucose in there. So we start killing that. Aldolase A has a lot of lysine and trisacine. The trisacine acts as an electron acceptor, and lysine and stabilizes intermediates, basically. Aldolase B, this is the liver type aldolase. This right here, oh, highly important in the liver, as you see right there. First picture is a real liver. If this does bug you, I do apologize, just don't look at it. But if you're, most biology students love looking at this stuff. There's your liver, there's a bigger picture right there. What your liver looks like. Aldolase B is in chromosome 9, so not even the same chromosome, it's a chromosome. And it helps with the big function of fructose breakdown and gluconeogenesis. Well, I told you, gluconeogenesis happens a lot in the liver, right? And also we break down with fructose, right? Hey, fructose 1-phosphate. We've seen that before, haven't we? We have not seen fructose 1,6-phosphate, right? I tricked you. We don't see that. It's fructose 6, not 1. So what happens is aldolase B takes that fructose 1-phosphate or F1P and it converts it into G3P so we can do stuff with it and either go up or down the chain if need be. That's one of its main key functions. Other side is also helping with glycolysis. It's like gluconeogenesis. But that fructose breakdown is very important because if you eat fructose and you don't have this enzyme, hereditary fructose intolerance, you're intolerant to fructose. Well, what happens is you make this fructose 1-phosphate or F1B, and if you can't break that down, that gets stored somewhere in the body. That takes a phosphate. Remember, ATP is your money, right? ATP is good money. ADP is 1-loss phosphate. That is bad money. That can't do anything in your body. So it took a phosphate to do that. You can run out of phosphates. That phosphate is gone forever if you're not getting rid of it. It's gone. If you lose a phosphate, issues can occur, right? You lack the ability to move muscles again. Everything starts slowing down, gets sluggish. Also is very toxic without it. And if you have this condition, it's recessive and it's very rare. But if you do have this condition, you just don't eat fruit with fructose in it or sucrose. You just only can eat glucose foods. But that's what aldolase is. Aldolase C, it's encoded. It's on chromosome 17. Don't worry about the hippo. Talk about it a little bit. Expressed heavily in memory and in Purkinje fibers and that. So aldolase C is going to be found in brain, smooth muscle, and neurotissue. So the reason why the hippo up there is the hippocampus. A lot of students have a hard time remembering what hippocampus is. The way I remember it is hippos have great memories, which that should be a joke to you. If you know hippos have bad memories, it's elephants. That's a joke. Explaining a joke makes it not funny. But now it's going to stick with you because that lame joke's in your head. The hippocampus can help with memory, right? So all the aldolase, all the energy you need to think of short-term, long-term memory, to activate those neurons there, that takes aldolase C into making glucose like that. Purkinje fibers, those are the fibers that make your ventricles contract. That takes energy to make those do that. Also, anything in the brain and smooth muscles, I'll take that. Now, aldolase C and aldolase A do work with each other. So a lot of scientists think that aldolase C is a byproduct, it's a fail-safe. If you have an issue with aldolase A, C can take over. If not, interestingly enough, because it's the main aldolase in the brain, neurodegenerative diseases like schizophrenia and Alzheimer's can impact aldolase C, and you can go too crazy or shut them off completely. So transcription factors. So what is a transcription factor? That is basically, it's a protein, basically a protein that controls the rate of transcription or DNA to RNA. It can attach to an RNA molecule and either turn on or off it, basically. So we have basically right here, we have activators or repressors. If an activator bonds, it's going to go forward. If a repressor bonds, it's going to stop it, basically. So that's what that means. Why are we talking about that? Well, because we can have these factors. So heptocyte nuclear factors, or HNFs. Hepto means liver, cyte means cell. So there's a liver cell nuclear, and nucleus means nucleus. So these are heptocyte nuclear factors. These are transcription factors that are going to control or regulate the amount of liver proteins, mainly aldolase, right? So we have HNF1, A, and B, and these are found in many cells. If they're found in the pancreatic cells, it's going to help with insulin. Insulin is very important in glucose, maintaining glucose, which helps in glycolysis and gluconeogenesis. It's going to take glucose and shove it into cells. So insulin does. In the liver, though, HNF1, A, and B is going to basically act as a tumor suppressor. What that means is your liver is always growing. But the problem is when cells are always growing, high risk of cancer happens. Well, these things right here basically watch, and if a liver cell is turned cancerous or into a tumor, it kills it off real quickly. But these will help control the growth of this. We then have HNF3. We have alpha, beta, and gamma. That's what the Y stands for. It's gamma. And these are transcription factors that help make albumin. It helps with blood osmolarity. Talk about that later on. And this thyroid hormone. Thyroid hormones are very important. These control metabolism. An increase in T3 and T4, thyroid hormone 3 and 4, increase metabolism. Start breaking down way more sugar. Starts breaking down way more things. More sugar is made. That's really good for that. Then we have this CTATT enhancer binding proteins, or C3b. They're also transcription factors. They can be found in heptocytes, liver cells, adipocytes, fat cells, hepatotic cells, blood cells, spleen, kidneys, and blood tissues. It's found everywhere, basically. But it can help with cell proliferation, differentiation, metabolism, and immunity. And these basically are going to be transcription factors, increase and activate the functions of these things. Really important is in the liver, it's going to help the liver make the proteins it needs to do its job, like gluconeogenesis and be detoxifying things and grow real quickly. And the kidney as well helps with gluconeogenesis. Here are my references. Hopefully you enjoyed the presentation. Thank you very much. Have a great day.